I want you to look down at your hands for a second and, just think about the last time you got a simple paper cut. Or even just remember the last time you caught a common cold.
Right, something we all just kind of brush off.
Exactly. You probably just grabbed a tissue or a bandage and went right on with your day.
Mhm.
But right beneath your skin, completely out of sight, there is this incredibly dramatic, highly coordinated biological cascade that was just kicking off.
Oh, it is an absolute microscopic war zone down there. And I think we tend to take the sheer mechanical complexity of that response entirely for granted, right up until it fails us.
Yeah, which is exactly why today's deep dive is taking us straight into the trenches of your own bloodstream. We are unpacking this massive stack of anatomy and physiology literature, and it's all focused entirely on your blood's frontline defenders.
So we're talking about white blood cells, or leukocytes, and platelets, which are known clinically as thrombocytes.
Right. And our mission today is to figure out exactly how these microscopic entities manage two of the most critical jobs in the human body: fighting off pathogenic invasions and sealing up vascular breaches.
Because if we step back and look at the broader medical consensus for, well, the last century, really, we usually treated those two jobs as fundamentally different departments.
Like fighting infection over here, stopping bleeding over there.
Exactly. You have your immune system and you have your hemostatic or clotting system. But as we synthesize these sources today, we're going to see a reality that is far more blurred. These systems actually function as a single interdependent network.
It's kind of like think about the blood flowing through your veins like a bustling city highway system. The red blood cells are your civilian delivery trucks. They just keep their heads down, hauling oxygen to the tissues, minding their own business.
Yeah, just making their deliveries.
Right. But the white blood cells and the platelets, those are your emergency responders. The white blood cells are the military, actively hunting down threats, and the platelets are the combat engineers. They're frantically patching up the infrastructure so the highways don't just collapse while the military does its job.
That's a great way to visualize it. And to really appreciate how these emergency responders function together, we kind of have to rewind to how they were first identified in the first place.
Because that changed everything, right?
It did. Finding them fundamentally altered how human beings understand and diagnose disease. It moved us away from, ancient philosophy and into modern cellular pathology.
Yeah, because before the 1840s, medicine was largely tied to humoral theory. The sources talk about this. Disease was viewed as an imbalance of abstract bodily fluids, blood, phlegm, black bile, yellow bile.
Yeah, diagnosis was largely just observational guesswork at that point.
So what changed in the 1840s?
Well, the turning point comes with a French physiologist, Gabriel Andral, and a German pathologist, Rudolf Virchow. Independently, they started looking at blood from certain sick patients and noticed something physically anomalous.
Like anomalous how?
The blood wasn't its usual deep red. It was abnormally pale, almost milky, and incredibly thick.
Wow. Milky blood. That sounds terrifying.
It is. And in 1845, Virchow examined a patient's blood under a microscope and saw it swarming with these colorless cells that were massively crowding out the red blood cells. He coined the term leukemia, which basically translates to white blood.
Which we obviously know today as leukemia.
Right. And the paradigm shift here is profound. He isn't looking at a patient's symptoms and guessing about their humors anymore. He is looking at the cellular composition of the blood and identifying the disease physically.
Right. You can actually see the problem.
Exactly.
Yeah.
It established that microscopic cellular analysis could yield a definitive diagnosis. But the funny thing is, Virchow didn't actually know what those white cells were supposed to be doing in a healthy person.
He just knew there were too many of them in a sick person.
Precisely. The realization that they were an active defense force came decades later, in 1882, thanks to a Russian-French zoologist named Elie Metchnikoff.
Okay, I loved reading about this in the sources. Metchnikoff's starfish experiment. It is just a brilliant piece of biological deduction.
It really is.
So he was studying these transparent starfish larvae in Italy, and because they are transparent, he could observe their internal processes in real time under a microscope. So he takes the tiny rose thorn and introduces it directly into the larva's body.
A remarkably simple manipulation, but it yielded a monumental discovery.
Yeah. He watched under his microscope as these mobile cells inside the larva immediately swarmed the rose thorn. They surrounded it and attempted to engulf the foreign invader.
And he coined the term phagocytosis, meaning cell eating.
Right. And he deduced that this is a universal biological mechanism. He proposed that human white blood cells do the exact same thing to bacterial invaders.
which is a beautiful leap of comparative anatomy. Yet the reaction of the scientific community at the time was overwhelmingly hostile.
Which, I mean, I found that pushback fascinating. Seeing cells actively swarm a foreign object seems like pretty definitive proof of cellular defense to us today.
Right, it seems obvious now.
But his critics were like, well, seeing them swarm a thorn doesn't inherently prove they are the primary defense mechanism against microscopic pathogens.
Yeah, that opened the door for his critics. And his main critic was a brilliant scientist named Paul Ehrlich. Ehrlich firmly believed in humoral immunity, not the ancient humors, but, modern biochemistry.
Okay.
He argued that the body defended itself using chemical compounds dissolving in the blood. What we would later identify as antibodies, Ehrlich's camp dismissed Metchnikoff's cellular theory, basically arguing that white blood cells were merely scavengers.
Like just cleaning up the biological debris after the chemical antibodies had already neutralized the threat?
Exactly. So you had this massive ideological rivalry, the chemistry camp versus the cell biology camp.
But the resolution to this debate is such a profound lesson in scientific humility, because they were both right.
They were. In 1908, Metchnikoff and Ehrlich shared the Nobel Prize in Physiology or Medicine.
Which is amazing.
Yeah, they proved that human immunity requires both the cellular arm, so the phagocytes engulfing pathogens on the ground, and the humoral arm, which are the antibodies circulating in the fluid, tagging targets for destruction.
You absolutely need both arms of the system. So Metchnikoff proved white blood cells are the defenders. But looking through our modern sources, calling them white blood cells is essentially a historical umbrella term.
Right, completely.
It's not a single type of cell. It encompasses 5 highly specialized, functionally distinct military units.
And the way we still categorize these 5 units today is actually a direct artifact of 19th century chemistry.
Which is crazy to me. We live in the era of genomics, yet medical texts still classify these cells based on how they reacted to Romanowski-type stains over a century ago.
I know, it's wild. But the Romanowski stain relies on this elegant chemical interaction. It uses an acidic dye, eosin, which is red, and a basic dye, methylene blue.
Okay.
So when you smear blood on a slide and apply this dye, the chemicals bind to different internal cellular structures based on their pH. If a cell contains internal sacs of chemical weapons that pick up the dye, they appear under the microscope as little granules.
And we call those granulocytes.
Exactly. And if they don't, they are granulocytes. It's a functional snapshot of their internal chemistry, not really a genetic family tree.
Okay, so looking at this army, the largest unit is the neutrophils. These are your short-lived, highly mobile first responders like the shock troops.
Very aggressive.
And for decades, we thought their only tactic was phagocytosis, just eating the bacteria like Metchnikoff saw.
Yeah.
But in 2004, researchers at the Max Planck Institute discovered this mechanism called netosis.
Right.
And it is arguably one of the most violently effective defense strategies in human biology.
Netosis is a spectacular act of cellular self-sacrifice.
Wait, explain how it works, because the source material was mind-blowing here.
Well, when a neutrophil is totally overwhelmed by pathogens, it radically alters its own internal structure. It actually decondenses its chromatin.
It unwinds its own DNA.
Yes, it unwinds its own DNA and then violently expels that genetic material right out of the cell. This creates a physical, sticky web in the extracellular space, heavily studded with antimicrobial proteins.
To trap and neutralize the bacteria.
Exactly. The cell weaponizes its own genetic blueprint to form a trap, dying in the process to ensure the pathogen cannot spread.
I mean, that is literally like Spider-Man deploying a web, except the webbing is woven from his own unraveled skeleton. It's so metal.
That's a very vivid way to put it, yeah.
So beyond the shock troops, we have the lymphocytes.
Yeah.
These include your T cells and B cells. If neutrophils are the brute force, lymphocytes are like the intelligence agency overseeing adaptive immunity.
Right. They are the reason a childhood chickenpox infection grants lifelong immunity. But they don't operate in a vacuum. They rely heavily on the third unit, which are the monocytes.
Okay, and what do they do?
Well, monocytes circulate in the blood, but when they enter tissues, they differentiate into macrophages, which translates to big eaters.
So they act as the heavy cleanup crew.
Mm-hmm.
But their critical function isn't just waste removal, it's intelligence gathering, right?
Exactly.
When a macrophage consumes a pathogen, it doesn't just digest it. It takes specific peptide fragments of that destroyed invader and actually presents them on its surface to the T cells.
Yes, antigen presentation.
It's effectively handing the intelligence agency the exact molecular profile of the enemy.
It is the crucial bridge between the innate, immediate immune response and the adaptive, long-term memory response.
Amazing.
And finally, rounding out the five, we have the eosinophils, which specialize in attacking multicellular parasites, and the basophils.
And basophils are central to allergic reactions. When they're triggered by something like pollen, they release histamine. And the histamine mechanism is fascinating.
It really is.
It forces the endothelial cells lining your blood vessels to contract slightly, creating these microscopic gaps in the vessel wall.
Right, so things can get out.
Yeah, it allows fluid and other immune cells to squeeze out into the tissue to fight the perceived threat, which is exactly why your nasal passages get so congested and swollen during allergy season.
So all five of these units combined determine your total white blood cell count. And in a healthy adult, the physiological reference range is strictly maintained.
And this brings up a really common misconception the sources highlight, this idea that more white blood cells equal a stronger immune system. People hear about boosting their immunity and just assume maximum cellular output is the goal.
Which is a dangerously flawed assumption. A count outside the reference range triggers immediate clinical investigation.
Right.
If it's severely depressed leukopenia, you are immunocompromised. But chronically elevated numbers, what we call leukocytosis, rarely indicate superimmunity. It frequently points to a myeloproliferative disorder.
Meaning what, exactly?
The bone marrow is malfunctioning. It's mass-producing defective, non-functional clones that crowd out the healthy cells. That uncontrolled proliferation is precisely what Virchow observed back in 1845.
So it's a structural malfunction, not a biological upgrade.
Exactly.
Now, we've established how this immune army handles microscopic invaders, but all of these units rely on the blood vessels for transport. So when the physical highway itself is compromised, like the paper cut we mentioned at the start, we require the other emergency responder formed in the bone marrow, the platelets.
And it is worth pausing here to address a fundamental anatomical reality that surprises a lot of people. Platelets are frequently referred to as cells, but they are actually enucleate fragments.
Meaning they lack a nucleus entirely.
Completely. They have no DNA of their own.
It's incredible that an entity completely lacking a cellular headquarters can execute such complex, receptor-driven cascades. They're essentially just microscopic chips of cytoplasm that bud off from these massive precursor cells in the bone marrow called megakaryocytes.
Right, just floating as cellular shrapnel waiting for a vascular breach.
But because of their size and their fragility, the medical establishment dismissed their physiological importance for decades.
Oh, completely ignored them.
In 1882, the exact same year Metchnikoff was studying his starfish, an Italian pathologist named Giulio Bizzozero used intravital microscopy to observe living blood vessels in rabbits.
Yes.
And he witnessed these tiny fragments rushing to the site of an injury and aggregating to plug the hole before the red blood cells even arrived.
Yet his peers largely dismissed them as staining artifacts or just random white cell debris.
Because the chemical fixatives used on slides in the 19th century often destroyed the delicate platelet membranes entirely, right?
Exactly. So researchers just assumed they were looking at slide contamination. It required advancements in vital microscopy to actually vindicate Bizzozero. Platelets are the central heroes of hemostasis, which is the process of arresting bleeding.
And the sources detail how hemostasis is not just a singular event, but this rapid multi-phase cascade. Let's walk through that.
Sure. Phase 1 is primary hemostasis, and this occurs in seconds. The damaged blood vessel physically constricts to reduce local blood pressure. Then, the circulating platelets detect subendothelial collagen that's exposed by the tissue tear. They bind to it using a crucial anchoring protein called von Willebrand factor.
And once anchored, the platelets undergo this dramatic structural activation. They morph from smooth, circulating discs into irregular, spiky spheres with these extended pseudopods, drastically increasing their surface area.
Right. They get incredibly sticky.
And they release chemical signals to recruit more platelets, aggregating to form a primary hemostatic plug.
So if the vessel breaches a broken dam, primary hemostasis is the city engineers frantically throwing sandbags into the breach. You need a sufficient reserve of platelets to manage daily microtraumas.
Right. The sources say dropping below a critical threshold puts a patient at risk of spontaneous, unprovoked bleeding. But a wall of loosely aggregated sandbags isn't structurally sound against arterial pressure for very long.
Which necessitates phase 2, secondary hemostasis. This is the coagulation cascade occurring over several minutes.
And this is where things get really complex.
Very. It involves a sequence of clotting factors mostly synthesized by the liver, and they activate each other in this amplifying enzymatic chain reaction. The cascade culminates in a massive burst of an enzyme called thrombin.
And thrombin acts as the biological catalyst here. It takes a soluble protein circulating in your blood called fibrinogen and cleaves it into insoluble fibrin monomers.
Right.
These monomers rapidly polymerize into long, tough strands.
So if primary hemostasis is the sandbag, secondary hemostasis is weaving a dense, unbreakable cargo net of heavy ropes around and through those sandbags, anchoring them permanently against the flow of blood.
The fibrin net locks everything down into a stable thrombus. And then once the underlying endothelial tissue has actually healed, phase 3 engages. Fibrinolysis.
Yes.
An enzyme called plasmin acts as molecular scissors, systematically cutting the fibrin ropes, dissolving the clot, and restoring normal hemodynamics in the vessel.
It is a brilliantly balanced system, but it also highlights the fragility of biological cascades. The sources use hemophilia to illustrate the devastating consequences of a missing component.
Right. Hemophilia is a genetic deficiency where a patient lacks just one specific clotting factor in the secondary cascade, typically factor 8 or IX. Queen Victoria was famously a carrier, passing the mutation through European royal lineages.
And without that specific factor, the entire enzymatic amplification halts. The thrombin burst never occurs.
So the engineers pile up the sandbags, but the cargo net is never woven to secure them.
Exactly. Consequently, even minor, unnoticeable internal injuries can result in life-threatening hemorrhaging because the primary platelet plug simply cannot withstand the hemodynamic pressure alone.
Now, for the last century, clinical medicine essentially kept these systems segregated. White blood cells handled pathogenic defense in one chapter, platelets handled mechanical breaches in another.
But the boundary between immunity and hemostasis is an illusion. The modern paradigm shifts the focus to how deeply integrated they actually are.
Yeah, modern immunology recognizes that platelets are far from passive structural bricks. They possess toll-like receptors, or TLRs, directly on their surface.
Which is huge.
TLRs are pattern recognition receptors. They allow the platelet to identify highly conserved molecular signatures present on the surface of bacteria.
Exactly. When a platelet detects a pathogen via its TLRs, it actively releases antimicrobial peptides into the bloodstream. Furthermore, activated platelets physically interact with neutrophils.
Oh, wow.
Yeah, they guide them to infection sites and actively stimulate that mitosis process we discussed earlier. The prevailing scientific consensus now views platelets as bona fide immune cells, functioning within this integrated framework known as immunothrombosis.
Okay, so if the clotting system and the immune system are essentially operating as a single unit, there must be clinical scenarios where this integration becomes pathological. Like what happens when the communication between these networks breaks down in a patient?
The most extreme manifestation of this collision occurs in severe systemic infections, like sepsis. It results in a terrifying clinical condition called disseminated intravascular coagulation, or DIC.
And the underlying mechanism of DIC is just a horrific biological paradox. In a state of overwhelming systemic inflammation, the immune system signals the endothelial cells across the entire vascular network to express tissue factor,
Which effectively tricks the body into thinking there is catastrophic vascular damage everywhere at once.
so the coagulation cascade goes into unconstrained overdrive. The body begins forming millions of microscopic fibrin clots throughout the entire circulatory system, even in the complete absence of any physical injury.
Right, and it systematically consumes the entire physiological reserve of platelets and clotting factors.
It uses up all the sandbags and all the fibrin ropes to build useless blockades across the entire city.
And because that consumptive coagulopathy exhausts the supply, the patient paradoxically begins to hemorrhage. They bleed spontaneously from IV insertion sites, mucosal membranes, and minor abrasions.
Because the blood is entirely depleted of the components necessary to form a functional clot where one is actually needed.
Exactly. It dismantles the intuitive assumption that clotting and bleeding are opposite ends of a spectrum. In DIC, a patient is tragically dying from systemic microvascular thrombosis and catastrophic systemic hemorrhage simultaneously.
It perfectly illustrates the dangerous intersection of immunity and hemostasis.
And you know, this complex physiology isn't restricted to critical care units. It dictates everyday clinical diagnostics, too.
Right, like the complete blood count or CBC. It is arguably the most ubiquitous laboratory test performed globally. When they draw your blood during an annual physical, the laboratory is fundamentally conducting a census of these exact leukocyte and thrombocyte populations.
Yes.
And a really relevant clarification for the sources here, leukocytes are not literally white.
No, they are completely colorless. They lack the hemoglobin that gives erythrocytes their red pigmentation. However, if you place a tube of whole blood in a centrifuge and apply high gravitational force, it separates by density. The heavy red blood cells pack into the bottom, the yellow acellular plasma rises to the top, and suspended right between them is this microscopic whitish-gray band containing all the leukocytes and platelets. It was termed the Buffy coat, which is actually the historical origin of the white cell misnomer.
The everyday application extends to pharmacology as well, right?
Yeah.
Tens of millions of adults take a daily low-dose aspirin for its prophylactic antiplatelet effects, to mitigate cardiovascular risk. Yet for decades, physicians were prescribing it blindly, entirely unaware of its actual mechanism of action.
It was a massive empirical blind spot. Clinical observation proved aspirin prevented arterial thrombosis, but the pharmacology was opaque until 1971.
When a British pharmacologist named John Vane discovered that aspirin irreversibly inhibits an enzyme within the platelet called COX-1.
Right. And by inhibiting COX-1, the platelet is biochemically prevented from synthesizing a signaling molecule called thromboxane A2.
And without thromboxane A2, the platelets cannot recruit each other. They cannot undergo the structural changes necessary to aggregate and form that primary plug.
It chemically revokes the platelet's ability to stick together. Vane was actually awarded a share of a Nobel Prize for uncovering that exact mechanism.
Wow.
But alongside these diagnostic and pharmacological triumphs, the sheer sensitivity of our modern technology has introduced new clinical dilemmas. The sources highlight the rising challenge of incidentalomas in primary care.
Yeah, because automated flow cytometers are now so remarkably precise that routine CBCs frequently flag these marginal abnormalities in a patient's white blood cell count. A count might be slightly elevated just due to acute physiological stress or smoking, or a minor asymptomatic viral infection that the adaptive immune system is actively resolving.
And this presents a significant diagnostic dilemma for physicians. Do you aggressively pursue a marginally elevated leukocyte count?
Right, because doing so subjects the patient to expensive, highly anxiety-inducing diagnostics, potentially culminating in an invasive bone marrow biopsy, only to confirm a benign etiology.
Conversely, dismissing the elevation runs the risk of ignoring the earliest hematological whisper of a developing leukemia. It demands nuanced clinical judgment well beyond simply reading the automated cytometry printout.
It really brings our discussion full circle, demonstrating the evolution of our diagnostic capabilities. We transitioned from Virchow's crude microscopic observation of sick blood to Meshnikov identifying the cellular defenders, to Ehrlich elucidating humoral chemistry, and Bidzo recognizing the hemostatic necessity of platelets.
It is a profoundly intricate web of protection. The entities constantly cycling through your vasculature, the highly specialized immune units, and the enucleate hemostatic fragments, they are the underlying biological reason we survive the daily microscopic hazards of human existence.
Their integration forms the absolute bedrock of modern hematological medicine, which leads to a final, slightly provocative thought I want to leave you with today.
Well, I like these.
Consider the historical progression we just traced. Medical science spent decades dismissing platelets as staining artifacts or worthless cellular debris, purely because 19th century microscopy lacked the resolution to observe their function without destroying them.
Yeah, literally thought it was dirt.
And we remained entirely unaware that neutrophils weaponized their own unwound DNA into defensive nets until 2004.
Barely 2 decades ago.
Exactly. If our current definitions of human biology are ultimately constrained by the limits of contemporary technology, what other invisible physiological systems, or what other supposedly worthless debris currently circulating in your blood are we completely misclassifying right now?
That is a fascinating question.
What critical life-saving mechanism is actively operating beneath your skin right now, just waiting for the next leap in microscopic resolution to reveal its true purpose? So the next time you get a simple paper cut, don't just put a bandage on it and walk away. Take a second to appreciate the unseen, fiercely coordinated biological network rushing in to save the day.